Okay, here we go. Okay. Hi, hello, and welcome. Microbehunter here again. Okay, everything set up now. Yeah, there's always usually around a minute delay when I press start, and then usually there is nothing happening for around a minute. That's why I have to wait a little bit. But I think everything seems to be working fine again. Hi, hello again, everyone. Welcome to another Saturday Microscopy live stream. I see that many of you have already joined in. Welcome all around the world. Thank you again for joining in. I know that some of you are joining in at what I would like to call impossible times. I know because of some time difference. Yeah, thank you in any case. Today I would like to put some blood under the microscope, a little bit of blood microscopy, also some blood staining techniques that I would like to try out, a little bit of theory as well. And I would like to invite you again to post questions in the chat. And after approximately 1 hour, I will try to answer them. So I will not answer them while I talk here, but I would like to kind of move all of the questions towards the end of the live stream. Okay, so to keep the whole thing a little bit more organized. I think the sound should work in any case. Yeah. And if you have any specific questions, microscopy related or biology related, please do ask them. It would make life easier for me if you would also include a little bit at Oliver or at Microbehunter at the front, because then the questions are a little bit easier to find. So I'm just going to start off right now, okay? And then as I go along, I'm going to then show you a couple of techniques. I'll also be taking some of my blood today as well, to also give you a little bit of a live view with a little bit of staining as well. So, but we're going to start off with a couple of permanent slides that I have here of blood, commercial slides. And I would like to show you a few of the things that you see here and explain a few of them here, okay? So what you see here in the background, this is now, we're using my 40 times microscope objective. This is a commercial permanent blood smear, as you call it, and what you are able to see here are, of course, the red blood cells. And there are a couple of things that I would like to explain here, okay? So each one of them are the red blood cells, and if you look very closely, you're going to see that they're not perfectly round, but they kind of almost fit together a little bit, sometimes like puzzle pieces. Okay, kind of like they're deformed a little bit. So maybe I go up with the magnification here so that you see this better. Okay, and there is a reason for that. Okay, so here we go. If you, for example, look at this one here in particular, you're going to see it's not completely round, but kind of, they kind of attach a little bit towards each other. And this actually already shows that the red blood cells are extremely flexible. And that they're able to, while they have a disc shape, they are able to deform a little bit. So that's one of the things that it becomes evident here. So this is actually an artifact. So an artifacts are all of those things and structures that you see in a microscope that are actually not supposed to be there, right? And this deformation is because the blood smear, as you see over here, is dry on a microscope slide. Yeah. And there's another thing that I would like to show you here on this permanent slide, and that is look at how the red blood cells are arranged. You see they're a little bit like pattern-like. They're kind of ring-shaped over here. There's some empty spaces in between here. And this is also something that happens during the drying process as the blood plasma starts to dry up and the liquid starts to retract. This basically the blood cells start to stick together like this, and then you have those basically places where there are these empty patterns in here, and we're not going to see those in a fresh blood sample. Okay, so this is basically something that you can not only see when you dry blood, but also, for example, last week when I dried some yeast cells as well, you could also see the same phenomenon. Okay, so these are basically two indicators here that we're dealing here with a prepared microscope slide. Here exactly. Yeah, here, bumped the camera here. We see it even more, right? That there are basically those empty spaces here, and that is because of the drying of the blood sample here. Yeah. So this is something that essentially we're going to see a little bit differently, and I mean, right now using my 60 times times objective. Okay, so if I remove the DIC filter, then this is in bright field, and you can actually see that it looks quite similar. We're also able to see a little bit the pinkish, the red blood cells actually look a little bit pinkish. Okay, however, there are a couple of other things I would like to show you here. Okay, how do those? This is now basically pure. Yeah, it's just blood, right? But we're also able to see some of those red blood cells in a cross-section of a tissue. Okay, so I'm just going to show you all of these permanent slides first before I actually go into the actual staining and so on. So I'm going to now replace the slide with another one. This one shows an artery and a vein. Okay. So basically, it's a histological cross-section. It's a little bit, we have to, okay. Maybe some of you already know this slide because it's not the first time that I'm showing this here, okay. So what we have here, a little bit of anatomy as well. It's a little too large, maybe. Yeah, here we go. It's a little too bright now. I have to refocus. Here we go. Okay, so that's basically now using my 40 times objective, okay. Looks like a chaos, but I'll quickly explain this to you. I've made separate videos exactly of this same slide. This one over here is the cross-section of a vein. You see it's it's kind of collapsed. Yeah? So you see that it's quite flexible here. And right now the inside here is white because there's not a lot of blood. But over here you see again a little bit of some of these blood cells here. Yeah. So it's basically you know what I'm just gonna show you. I'm going to roll up a piece of paper. Right? Yeah, if this is the vein, yeah, this is the vein, then it's kind of collapsed like this, right? So, and that's the reason why it kind of looks the way it does over here, right? And this one down here, that is the cross-section of an artery, and you see already that the artery has a much thicker wall, because arteries have to withstand a larger blood pressure because arteries carry blood away from the heart and veins carry blood to the heart. But we talk about the blood, and the reason why I show this to you is because I did find a few red blood cells here as well. Okay, I have to always go up a little bit yet again with the magnification and also the brightness. Okay, so here we're zooming in. That is the arm. Yeah, here is the artery again. So let's go up. Okay, forgot to do some of the adjustments of the microscope here. Let's go up with the magnification. And what you see in here, those dots here, these are also red blood cells. So the magnification is only now using my 20 times objective, so the red blood cells are much smaller. And they're also kind of sticking together. That's why we cannot see them as individual cells. Yeah. But just to show you a little bit the size difference here between the wall of an artery, yeah, and the actual red blood cells that we have in here. Yeah, so here as well. Okay. Yeah. And yeah, so that's basically something I want to kind of show you as well, simply to compare the size difference. I would like to continue now. There are two more slides I would like to show you. It's another interesting one here. That's again a commercial one. This is I kind of like this one here a lot because this one actually shows some parasites inside blood. We have to go up again with the magnification a little bit here. Okay. You know what? This is not quite well visible yet. So here we go. I remove the filter. So, and that's also blood. I'll explain this here again. If you look very carefully, you're able to again see the very brightly over here the red blood cells. And then do you see those dark worm-like structures? These are protozoans called trypanosoma. These are the parasites that cause sleeping sickness. And they are, basically carried by the so-called tsetse fly. And basically they all are in the blood and they reproduce in the blood. And this is a sample of basically these trypanosomas as they are in blood. Okay, so this is something, where when I saw this slide the first time, I said, wow, there are a lot of them here. Now, I don't know if this is actually a natural blood sample or whether the company who made the slide did not actually reproduce them and grow them inside blood, because the concentration of these trypanosomes is crazily high. Huh? But this also shows a little bit that with proper staining, you're able to make them quite well visible. Okay, and here evidently they use the stain that stains them darker, and the red blood cells, they are still quite pinkish and pretty bright in the background. And I think the concentration of those parasites here are pretty it's pretty high. Yeah. So these are by the way, if you're interested in this, you got to check this YouTube channel. I did some months ago, last year, or I don't know, I did actually make a separate video about those. Yeah, these are single-celled organisms, those trypanosomas. Yeah, I made actually a separate video about those where I actually showed you exactly the same slide. Yeah, but simply to show you a little bit on what level we are here. And then last but not least, I've got another slide here, and this one here is also one that I kind of like, because it kind of shows the difference, because this one is now, what I'm going to show you now is the blood of a frog. Okay, so basically of amphibian. So let me show you. Because there is an interesting and important difference here. Okay, here again, these are red blood cells of a frog. Let's go up again. And here we already see a very, very important difference. Actually looks a little bit, I would say probably a little bit better yet than the others. You see that there's a dark dot in each one of them. That's the nucleus. So the blood of frogs and of amphibians, they have a nucleus, and the blood of mammals, like us humans, they do not have a nucleus. So during the formation of the red blood cells, the nucleus was lost and the space is used for hemoglobin, which is the pigment that carries, the protein that carries oxygen, right? And in amphibians, the nucleus is still present, and here they stain it and you can still see it. Yeah? So basically, kind of shows a little bit the different strategies of actually, of carrying oxygen here. So evidently in an amphibian, in a frog, maybe they don't need as much hemoglobin, so they were able to retain the nucleus here. Huh? So this is kind of the idea here, simply to show you a little bit of blood here. If you just joined in, if you want to ask some microscopy or related questions, please feel free to do so, to type the questions into the chat. And after the first hour of the live stream, I will try to answer those questions in one session, so that I'm not interrupting myself. This at least has been the request by some viewers in the past, and I think it works quite fine this way. Okay, so I'm just going to do a presentation right now, and I'm going to ignore you, so to say, and then later on I will try to address those questions. Okay, so that's a little bit the idea. So what I'm going to do now is the following. I just realized that I actually forgot something. Ooh. I actually wanted to also show you what happens when you add salt water to blood. So I'll see if I'm able to do this as well. I forgot my salt water. So what I'm going to do now is I'm going to show you something scary. Because I'm going to now make I'm going to first remove those slides here, because I do not want to get them covered with blood. Okay, so what I'm going to do now is I'm going to show you something scary. Something that I bought some time ago. These are stair lancets. They look like this, and basically they're used to prick yourself to draw some blood. I say scary because they're scary, right? It's a lot of psychology. There's a protective cap here, and you have to prick yourself, and I'll press some blood out, and I'm going to put it under the microscope. Because I want to show you how this actually looks like. Because actually, as a matter of fact, the blood, fresh blood, looks much nicer and more interesting than what I just showed you. And this is actually also one of the reasons why I showed you those permanent slides, so that you're able to see a difference between the permanent slides and the fresh blood, so to say. Okay, so what am I going to do? I'm going to take a slide. We're just going to look at some fresh blood first. Then I'm going to stain some of the blood using methylin blue. Many of you already know this solution. I've been using it quite frequently. And this has the advantage that we're able to see the white blood cells better. And something I've not tried. Okay, because I want these live streams also to be a little bit, yeah, experimental. So maybe it's not going to work. I've got some fountain pen ink here. Okay, you can also buy it in cartridges, and yeah, it's basically used by lots of school children. If you're from a German-speaking country, you probably already know the company. I will have to say it's not sponsored, obviously. Yeah, but I bought myself some time ago this bottle here because it's, yeah, I use it also as a stain, and as a matter of fact, you can stain. It does work, but I've never tried it with blood before. So maybe it's not going to work, okay? So this is kind of the thing that I just want to show you. So what I'm going to do now is I need a blood sample. And now this is where it starts to become a little bit stressful for me, okay? Because I have to hurt myself now. And before I do that, I would like to use a little bit of alcohol simply to disinfect my finger, because that's what you're supposed to do. So I've got this bottle with alcohol, and yeah, I will simply use this alcohol now to disinfect myself a little bit. The alcohol will do the following. It will kill off bacteria that are found on the skin. Why will the alcohol do that? Because the alcohol will dissolve the cell membrane of the bacteria, and this way the bacteria pop open and yeah, die. And that's how basically alcohol disinfects. Okay. Soap, by the way, does something similar. I think that's definitely enough. And now this is where the tough part starts. Okay. Okay, I'm going to show this to you now. Look, there is a little yeah, cap on here that you have to remove because it is sterile. Yeah. So look, you remove this, and this is how it looks like. There is a sharp needle here. Okay. And what I have to do is I have to prick myself, and this is really psychologically difficult to do. Okay. So what I will do is I will try a different way. I will try it simply like this. Okay. It's easier this way. It only hurts at the very beginning, and yeah, there are lots of capillaries. There are lots of blood capillaries here in the fingertip. Yeah, turning it also hurts a little bit, but this actually means that maybe more blood vessels break up, and here we go. Ta-da! Am I happy? What I do is I apply a small drop of the blood here on the corner. Not on the corner, but on the side. And I'm not going to make a blood smear, okay? So normally, if you're in medicine, what you do is you now take another slide, okay? And you kind of you streak it across, yeah? I'm not going to do that because I do not want the blood to dry out, but rather I'm going to now add a fairly large cover glass. Because what I'm going to do now is I want to have a very thin film of blood directly beneath the cover glass. Yeah, and because there's a cover glass on top, I think I'm just going to wipe this again here. Because there is a cover glass on top, it's not going to dry out as quickly, and that's actually what I would like to do. Okay. Yeah. Thank you very much. This is also something I would like to invite you to do. You can, yeah, contribute a little bit financially, and what I've done already, because thank you again, some people last week donated a little bit of money, and I've used this money to buy some microscope slides, which I would like to show you when they arrive. Okay. Yeah. So this is simply something that I wanted to, yeah. Also mention here. So I'm going to put this now under the microscope, and we can actually see that the blood is now slowly moving across due to capillary action beneath the cover glass. So let's have a look. Okay. And then I guess I have to prick myself multiple times today again. Okay. So, and let's see the scope view again, and we have to, of course, turn up the light. Here we go. Okay. So, and that's actually one of the nice things, because yeah, there is look. We just ignore that. There are a couple of some people say, "Oh my gosh, what is this here? These long things here." Well, this is just dirt on the microscope slide. Yeah. So there's nothing to worry about. Yeah. Those large things here are, of course, bubbles. Yeah, and what we see is we see that the blood is now due to capillary action starting to spread. Yeah. Again, you will see some. The reason is I cleaned the cover glass and the microscope slide with some tissue paper. So these are probably some fibers some fibers of cellulose fibers of the tissue paper. So we just ignore these things. Yeah. Yeah. And the density is quite high, but where is this? Here the blood did not go yet. Yeah. Yeah. But it's slowly spreading. Okay, so, and then I'm going to show you a couple of things here as well, which cause some people to feel a little bit worried, but there is no cause. Okay. Yeah. So, you will actually see that many or some of the blood cells, well, let's try to go to, here it's better. Yes, beautiful. This is called rouleau formation. Do you see? And sometimes people freak out when they see that. But there's nothing to worry about. Do you actually see the red blood cells stacking up? Yeah. So this is basically it's called rouleau formation. It's something totally normal when blood starts to coagulate because the red blood cells have a disc shape. For this reason, when they glue together, they like to stick together, and sometimes they like to aggregate by essentially stacking up like a disc of coins. Yeah? So that is actually something quite normal, and sometimes this depends also a little bit on the amount of blood that you're using, because sometimes if there is a lot of pressure on the cover glass, then essentially they're not able to stack up quite as easily. Yeah. But that is essentially, let me move over here as well. Yeah. This kind of shows quite nicely the individual red blood cells. And I'm going to also show you then a video in time lapse. So now, if you are basically very, very patient, and this is possible, but it does require a lot of patience, you might be occasionally able to see also some white blood cells. Okay, and sometimes they move around as well. Let me turn off the arrow. Okay, and because they are sometimes difficult to see, and because sometimes they move slowly, I've made a time-lapse video of this as well. So you might, I don't know, look, this could be one. I'm going to turn on the arrow again. I don't know if you're able to see this. But for example, have a look at this one over here, right? Yeah, this might be, if you see that it does change slowly its shape, so this might be a white blood cell. There are so-called phagocytes in the blood, and what they do is they move around like an amoeba, and what they do is they remove dead cells and particles and basically in the tissue also bacteria and so on, and by engulfing it, yeah? And if you have to be really patient, and then you're able to see those, and you can now see that they are moving sometimes very slowly, and for this reason you have to really, if you find one, you have to really look at it for a couple of minutes, and now as you see it's gone, it probably disappeared somewhere over there with the other red blood cells. But I'm going to show you then a high magnification time lapse that I made. Okay, so but I just want to make it clear. Okay, over here for example, this one here, as well, could also be a white blood cell. Yeah? Maybe this one over here as well. So the red blood cells are significantly more, simply more abundant. Yeah. So yeah, and basically, what why not? Before I do some staining and some other things, why not show you the video? You see again here this huge thing here, yeah. It's a contamination. It's a dust or thing that was on my microscope slide. Yeah? So if you see those things in a blood sample, I know from forums, people see and they're kind of panicking. What in the world is that? I said, come on, it's nothing. Yeah. Look at this guy here. Maybe you're able to see this one over here. Yeah. Might also be a white blood cell. Not all white blood cells actually move. Yeah, so there are different types. You know what I would like to try now? I'm just going to try to press now against the. I'm just going to use just a second. I would like to press now against I would like to press now against the microscope slide to see how this is actually. Yeah, see when you press. Yeah, here. Yeah. You see how the blood. When you do that, you're also starting to break up a little bit the stacks, of cells. Yeah. So let's move over here to the side. Maybe there are fewer. Okay, because sometimes if there are fewer cells visible, then you might be. It's easy also to see the white blood cells. So if you go here to the side, sometimes, well, maybe not so much here, but if the layer of blood is very thin, then you're going to see that most of the cells will actually start to lie flat because they're compressed between the cover glass and the slide, microscope slide. So for this reason, they are not able to stand up, but they're kind of compressed flat. And then if you're very patient, and if the blood starts to dry out, and sometimes you see this on the edges happening first, then you might also see that they will start to, the blood cells start to develop spikes. And this is because the cell shrinks because it loses water. Yeah? So this is also something that, basically is sometimes visible, okay? So what I would like to do now is I'm just going to show you quickly the time-lapse video that I made of my blood and using a very high magnification. So I think I might have even used the oil immersion for that. And I was speeding it up 10 times. Okay, have a look at this here. Okay, so what we see here is 10, that's one of those white blood cells. And look how it's moving around. And the other red blood cells over here are simply moving around because of capillary action. It's another white blood cell down here. This one over here is called a ghost. It's a red blood cell that popped open and lost its contents, also up here. Yeah? Barely visible. Yeah? And you see that this, it's kind of moving around like an amoeba. So this kind of... This is actually quite fascinating to see. Yeah, so this is something where you have to be really patient. So the video is actually quite long, and then I was really going into time lapse and speeding it up. Yeah, and you see that they're not all white blood cells are actually moving around. The one down here, for example, is just sitting because they're different white blood cells. Yeah. So those I know on YouTube I've seen other videos as well where. Those white blood cells, those phagocytes, were actually able to follow some bacteria around because they're able to sense the chemical trail. Yeah? So they're actually able to follow, they're able to kind of, under quotation marks, maybe smell bacteria, and they're able to follow them, and that's how they're able to catch them down. Yeah, here in the top, is actually one of those cells that I was talking about. This is when the blood starts to dry up. It loses the blood cells lose water, and then the membrane will start to form wrinkles like this. Yeah. So the longer you wait, more of the cells will start to form these spikes. Yeah. You see already some of them down here also developing those spikes. Yeah? And this is a very, very thin layer of blood, and that's why all of the blood cells are able, basically are in one plane. Yeah? I've also tried to do this with a little bit more blood, but then it can happen that the white blood cells kind of move also vertically in and out of focus. Yeah. So, but actually, I think that's actually quite fascinating. Yeah. Yeah. So, yeah. So this here is in this here is in DIC indeed. So, but let's go back now to the let's go back to the regular scope view of what I've got here. Yeah? And that's basically. And if you want to observe this in bright field, then this is how it looks like in bright field. Yeah. And why not? I can also have phase contrast. Why not try also phase contrast? But then I need the phase contrast filter here as well. Need to open this, and this is how it looks in phase contrast. Actually, also kind of pretty. Yeah, but again, phase contrast only works best if the layers are fairly thin. Let me find it again. Ah, yeah, here. It's too thick. Actually, this blood is not spreading quite as nicely as I hoped it would. I think it depends also a little bit on. It's also quite interesting. The consistency of the blood depends also quite a lot on whether you have been drinking a lot of water just recently or not. Yeah? Remember, once I was at the doctor's, they had to take some blood from my earlobe to do some kind of an analysis, and they could not draw any blood. And then the doctor says you should be drinking a little bit more because, And then I had to drink a couple of glasses of water, and then they were able to draw the liquid again. Yeah. So to say and to show you that this also depends quite a little bit on how much water you drink. It determines also how thick or viscous the blood is. Here again, very nicely, we're able to see some of those stacks. Yeah. So let's go back here again. Yep, here we go again. Yep, so let's press against it a little bit and let's do a little bit of some other stuff. Yep, see? And when you do that, you actually see how the red blood cells start to deform. And then those stacks also start to break up, of course. It goes a little bit out of focus, of course, because I'm pressing down on the slide. Okay, so let's move on a little bit. Okay, so let's move on a little bit. I think I have to draw again a little bit of blood, and then I would like to do a little bit of staining as well, and then let's see how this actually works out. Let me show again the desk view. Let's the desk again, and let's see how much the blood has spread in the meantime. Yeah, so I don't know. So actually, sometimes in some cases, I've actually seen the blood spread even more. Yeah. So let's try it again. Let's move this somewhere else. Yeah, this is also clean. I have to. Where is this? Yeah. Today I'm going to hurt myself quite often. Okay. Okay, let's try it here. Let's take a little bit more. Okay, here we go. And I would like to now do the following. I would now like to do a methylin blue stain. And I do that by simply adding a little bit of so-called Löffler's solution. I know that for staining blood there are other stains that are more commonly used, but I was quite successful with this one. So I'm just going to stick to it. This is methylin blue, and methylin blue is able to stain DNA. Okay, so I'm going to add a drop of this here. On here. And what I found out, what is really important is you have to mix it properly. Okay, and I do that by simply drawing it in and out, because if you don't do that, then I found out that not all of the cells are properly stained. Okay, so I do this a little bit. This, of course, will kill the white blood cells. There is potassium hydroxide. There is also alcohol. See, that's actually not a good idea. Now I've got some foam bubbles forming. Okay, so what I'm gonna do, maybe I'm just going to take some of this here, like this, and maybe put it on a new slide without foam. Yeah, without. And cover glass goes on top again. I don't know, should I use a smaller one? I think I'm just going to use a smaller cover glass. This one over here. I have to clean it off a little bit. because so, and it goes on here. Okay, hope it's seen enough. Lots of bubbles again. I don't care. Let's put this one away. And yeah, let's give this one a try now. I have to change to scope view. Here we go. Okay, wow, okay, there's still some capillary action happening here, some movement. I have to wait maybe a minute or so for the whole thing to stabilize. And I think there are lots of cells. There are too many of them now. Yeah. You see, there are too many of them. That's why they're kind of overlapping a little bit, but I can already see that what I wanted to show you. But actually, it's better when there are when there are fewer cells. I've already seen some stained white blood cells. But, okay, I'm going to now do the following. I'm going to repeat this with a little bit less liquid. Okay, so I'm going to take this away here. I'm going to still have plenty of this here in any case. So let's give it another try. Let's take very small drop now. This one is unfortunately, this cover glass was broken. This one is better. Let's wipe it again. So, maybe this is going to work a little bit better. I hope that I'm getting a very thin layer, yeah, here, and let's give it another try. Oh yeah, I have to change over to the scope view here. I think that might be better. Okay. So yes, beautiful. Yeah, I'm going to move up to 60 times. And you know what? I'm just going to maybe going to put the filter away. Yeah, here we go. This is now basically bright field, and we can already see that I bumped into it. That some cells, like the ones over here, are stained blue. Okay, and these are white blood cells, but these are not the ones I wanted to show you, because some white blood cells have a very interesting shape concerning the nucleus. This one maybe is also not a very typical one. You also see right now that many of the red blood cells, they are not quite as visible anymore. Many of them probably popped open already and have lost the contents. So here are again 3 of them. Okay, and what you see is the following. You see that the cells here, the white blood cells, have, yeah, of course, a certain round shape, and then in the middle, it's darker, and that is the nucleus. And based on the shape of this nucleus, you can actually categorize different white blood cells. So but there are those, some of them they're called polymorph nucleated. And the nucleus has several lobes. Again, it's a contamination here. We just ignore that. Yeah. Ah, yeah. Here might be one a little bit. You can actually see that this nucleus over here, right, in the center is not completely round, but seems to have lobes. Yeah. And depending on whether it has a round nucleus or a lobed nucleus, you can basically distinguish different white blood cells. Yeah, here, for example, at the top, this huge blue thing on the left, we ignore. Yeah, this one, for example, you see that the nucleus seems to have here 2 lobes or 2 parts. Yeah. Yeah. So we basically ignore this contaminant. Here again, a white blood cell. Yeah. Again, you see that the nucleus seems to have be lobed. Here are a couple of white blood cells as well. And essentially by staining this, you can actually do a white blood cell count. Yeah, and many of the red blood cells, you see that they have basically completely lost their shape. Yeah. Here again. What I would like to do as well is I would like to also try the staining now with the ink. Yeah, and I don't know if this is gonna work, but yeah, here that's actually one of those that I kind of wanted to see. Do you actually see that here the nucleus is kind of like bent, kind of? Yeah. So that's also something that you sometimes are able to see, and sometimes you're even able to see that it's not just bent, but there are actually 2 or 3 lobes. Yeah. Polymorph nucleated granulocytes, I think they're called. Huh? A polymorph meaning having different shapes. Huh? And here again. So you see that actually making those white blood cells visible is actually quite fairly easy. Yeah? Here you see also that the white blood cells, the nuclei are different lobes. The reason why methylene blue stains the nucleus is because methylene blue has an affinity to DNA. It likes to stick to the DNA. And yeah, here for example, here in the corner down here, yeah, you see that this one over here seems to have 3 lobes. Yeah, 1, 2, 3. Yeah. So that is essentially something here again quite nicely visible. Here again, multiple lobes. Yeah, so it's kind of interesting to see that with the relatively simple techniques you're able to make the white blood cells visible. Yeah. Again, methylene blue I think is not the standard one that you use. There are different stains which are more commonly established for blood microscopy, but in my cases it worked well. Yeah. So let's try the following. What time is it? Okay, around 15 minutes. And then I would like to answer some of your questions. So what I'm going to do now is I have to again prick myself, unfortunately. And I would like to then try this with some fountain penning. Ah, look, here. Now you're actually able to see this. Sometimes it takes a couple of minutes for the stain, the methylene blue, to be properly absorbed. Yeah. But here you can actually make the white blood cells stand out, and the stain has also causes maybe the red blood cells to pop open and to lyse, and then you do not see them quite as well. Also allows you to see how many more red blood cells there are compared to white blood cells. It's also possible to actually also see the blood platelets, which are important for blood clotting, but they're significantly smaller. Here we go again. Yeah. So let's I'll prick myself again. Then let's try it with fountain pen ink. But again, this is something I've not tried myself yet, so I'm not sure if it actually works or not. Okay. So I put this away here. I put this away here. Yeah. Yeah, I have to do this again. Where is a microscope slide? Here it is. You know what? I'm just going to put a little bit of maybe ink out first. I'm a little bit worried that maybe it's going to be a little too dark. Up, that's a lot. I know that this ink works quite well in staining cheek cells. Yeah, so I know that it does stain cells, but I've not tried it with blood cells yet. So this basically means, again, I will use some tissue paper. To disinfect myself a little bit. So let's take this here again. It's easier actually flicking your finger than actually trying to push it in. Psychology again, you know. Lots of This also worked fine. You know what? Just for the sake of it, I'm just going to show you now how you could actually make a blood smear. I'm just going to demonstrate this as well. Again, yeah, how they do that. If you want to actually also stain it, you put a drop of blood on one side. You take a second. Microscope slide and do the following. Yeah, do it like this, and then you go. Okay, and then you allow this to dry. Yeah, and then you can stain it as well. Yeah, so I'm going to discard this. Okay, we can have a look at this as well later on. It might be still a little too dense. Yeah. So what I'm going to do now is I'm going to now use this here. Okay, I'm going to use this here now, and I'm going to add a little bit of this ink, not too much because I know that it's quite strong, and I do not want to make it too dark. So I'm going to mix this again. I know if you want to do this properly, you probably have to get the correct quantity of each and all these things, right? I'm not doing that. So a small drop here. I'm going to use a cover glass again. This cover glass is a little bit, okay. There are two of them, okay. Let's put it on here. And again, I don't know if this is how well this actually works, okay? So this is the first time, really, it's the first time that I'm trying a regular fountain pen ink to stain blood. Okay. And yeah, thank you. I just received your questions. Okay, so here are your questions. And let's have a look. Where is the scope view again? Here we go, here we go. Okay, here are the cells. Need to go up with the magnification. I'm going to remove the filter, hoping that this way I'm able to see the blue cells better, if they actually stain blue. Yeah, in any case, the ink does not seem to have impacted so much on the shape of the red blood cells. But if I look at this, I'm not seeing any blue stained. And the movement is, of course, because of the capillary action and the evaporation of liquid. Yeah, so, look, I don't know. This one looks a little bit bluish, but not really very well. At least I think we can agree that maybe this fountain pen ink might not be the best stain, at least in the way that I've done it right now, for blood. I mean, it does stain a few things blue. I mean, you see, I don't know, whatever that is. Yeah, either I did not use enough of it, but it actually did look quite dark. But I think we agree that probably the other one was better, and now they're really trying to stack up a lot as the blood starts to coagulate. Yeah. Okay, I think this kind of explains a little bit. Yeah, you know what? I'm going to try it in any case. I'm going to try a little bit more. I'm going to try to add a little bit more of the stain, and maybe. Maybe this does make a difference. I don't know. So Where is my cover glass? Oh yeah, no, not this one over here. I use these ones over here, still a little bit smaller. And let's try it here. Yeah, I see it's significantly more bluish. And scope view again. Let's remove the old slide, in goes the new slide. And focus. Higher magnification. So next higher magnification. Oh yeah. Here we actually see that because I added more liquid, they do not stack up anymore. Yeah, some contaminants that you see in here do stain a little bit bluish, but I don't know. I don't see. Yeah, these things. These are just blue things. Are just contaminants, but I do not see any white blood cells now. So I think the methylin blue from before was actually a little bit better, I would say. Yeah. So I would say that, I don't know, maybe, could it be that, I don't know. Yeah, I don't know if this is one or not. It doesn't really. You see, individual cases really don't count a lot. It could be any other contaminant. You have to see them more often to actually be sure that they are white blood cells. And the other things that you see here are too big. And yeah, okay, I think we come to the conclusion that probably the blue fountain pen ink is not the best stain to be used for staining blood, at least the white blood cells. Yeah. So, yeah, so that's simply something I wanted to show you. But I just made a the ones this one over here, the desk view here. Remember this one? Let's have a look at this. Okay, so this one is now basically a little bit air dried. Let's see how this looks like. And here it goes. And let's close the whole thing here a little bit. So that is now the air dried version. Let's go up. And it looks again different because of the difference in refractive index between. Oh yeah, it's actually quite interesting here. Yeah, usually that's what we see here. That's also quite nice to see. You see that in some cases that the borders or the edges of the blood cells are actually quite pronounced and darker than they actually are, and that's something that you see quite often when you have a specimen which is not surrounded by liquid medium, but which is in air. So the differences in refractive index are so large that sometimes you see those dark edges. Okay. You know what? I'll try now a phase contrast. And I have to open this here. This is a phase this is not phase contrast. It doesn't look very. Yeah. So what I'm gonna try now is the following. Also something I didn't do yet. And that is I'm going to now directly add a little bit of methylene blue stain on here, and let's see if that works. So before I mixed the methylene blue with the liquid, the blood, right? And now the blood is dry, and I would like to see how the staining works when I add the methylene blue directly on here. Oh, I bumped it again, and a small amount of methylene blue. And the way that I do that now is as follows. I'm going just going to, it's not very much. Okay, here, that's better. So I have to wipe. So let's give it a small drop here. Cover glass goes on top. Never tried this technique. And let's see how this works. Okay. And it goes again under the microscope. Okay, I have to remove the phase contrast filter. Just a second, yeah. Let's go up with the magnification a little bit. Okay. Okay, and yet up. And open the condenser a little bit. Let's remove this here. Let's go again into bright field. And 60 times, yep. Yep. Yes. Look. I think we can agree. Now the red blood cells are not visible at all, but the white blood cells are. Oh, it look interesting. Look, almost looks like it's dividing. Yeah. So this basically seems to work quite well. If you again those irregular things that you see in here, forget about those. Huh? And I think this even works even better now. Yeah. So I've dried the blood. First I made a blood smear. I dried it, then I directly added some of the stain on top. And what this has done somehow, it seems that the red blood cells are not visible at all anymore. Okay, so this maybe they have all lysed and broken open, yeah. But the individual the white blood cells are still there, and they have now been, their nuclei has now been stained. Those big things we ignore again. Yeah, and let's go down to decrease the magnification, and you now can see all of those blue dots here, which are white blood cells. Okay, just wanted to show it like this to you. Okay, so what I'm gonna do now is I'm going to I'm going to interrupt myself again now. Look at all those blue dots here. Yeah, and I've got I received a few questions from you which I would like to answer. Okay, so this is something that I would like to address now. And we're going to see how far we're able to go here. Yeah. So before I do that, I have to clean up everything a little bit again, because there is stain and blood and everything around here. And I do not want to, yeah. And of course, this lancet over here, this lancet over here, I have to put it back here, and I have to dispose of it carefully. Yeah. So I've got a separate trash can. And let's go through those questions. Okay, if you've got some last second questions and you would like to post them, please do so. I will at the end. I will also try to read through the chat a little bit and maybe wrap up any remaining questions that there are. Let me quickly remove the other things here as well, so that the microscope is reasonably clean. So here we go. I have not read them yet, so this is going to be a little bit of a new thing for me as well. I would like to thank again Michael, who has copy-pasted those. So, have you ever tried autofluorescent microscopy? Short answer, yes. I wonder if it will be interesting to make a video showing how to upgrade a simple compound microscope to dioscopic fluorescence. So I'm going to quickly explain to you what autofluorescence microscopy is, and techniques that you can use, and I have had also some success with it. when you use autofluorescence microscopy, what you're doing is you're using not necessarily ultraviolet light, but also very deep blue light also works. And when you shine blue light on some, let's say, green cells, chloroplasts, for example, inside plant cells, algae, for example, and when you are using a dark field filter at the same time, then they will start to glow red, very weakly red, but they will start the fluorescence to glow. And you can actually capture this with a camera, with a long time exposure. So this basically is in principle working. I also was partially successful with that, but it wasn't as impressive that it wouldn't be worth actually making a video out of it. Yeah? So that is one thing. And number two, I've also experimented a little bit with so-called UV flashlights. I've tried to upgrade a microscope also using UV or blue light LEDs. When you do that, then you need a dark field patch stop to make sure that the blue light or the UV light only shines from the side. And then you might be lucky that you're able to see some of the cells actually start to glow. Yeah? So yes, I've had a little bit limited success. Yeah. So where do you get these things? Unfortunately, I don't know now what this refers to. Okay. Yeah, other veins, or maybe you're referring to the slides that I have here. Those commercial permanent slides can be bought. Okay, you can even buy them on Amazon. The quality varies. And those slides are made by companies which also make them for medical training. Okay, so sometimes some of the slides can be quite rare, so to say, showing very rare specimens or even diseases and so on, parasites. Yeah. So these slides, if you're referring to the slides, can actually be purchased. Okay. Other veins actually blew outside the body. I thought the color was imbued by the deoxygenated blood. As a matter of fact, the blood is still, let's put it this way, the blood is a little bit darker, but it's not really blue. It's a little bit, the reason why sometimes veins appear to be blue is also because of the, not because the blood is blue. Right, it has to be because the blood is indeed a little bit darker. I think deoxygenated blood is a little bit darker, but it is still red. Yeah. So, and in the case of the slide, this was because it was stained. Yeah. Wouldn't it be interesting to mention the objective magnification used with your images? I know it's not the actual magnification, but this way the hobbyist has a guideline. So yes, and this is the whole thing with the objective is a complicated matter. And this has, I would like to explain this to you a little bit more detail. Yeah, I've switched off the microscope now. Okay, but I would like to explain this a little bit because this is actually an issue. Okay, so what you see up here at the top is basically the objective. Yeah, it's a 4 times objective, and I'm just gonna show you maybe just the slide again with the artery and the vein. So just that you kind of, let me show this to you. Yeah, see, it's a little bit, So, okay, let's just focus this a little bit. So, and I would like to illustrate now something here. What you see over here now with a 4 times objective, how much you see from here to over here, right? This is referred to as the field of view. And this field of view is different from microscope to microscope and from camera to camera, and it depends also on the size of your camera sensor. So the thing is the following, that if I tell you right now that I'm using a 4 times objective, this does not necessarily mean that you're going to see it the same way. It could be that in your microscope, because you have a smaller camera sensor, that you're maybe only able to see a smaller region from the center, right? But that magnified more. Yeah. So this is a little bit the problem of actually comparing magnifications, because if I say, well, I'm using a 4 times objective right now, it does not mean that you're able to see it the same way. Yeah? And this basically makes it very difficult. Now, if I were to, and some people have suggested this, why don't you just write down 4 times here on the corner? but that is again misleading, because if I write 4 times here in the corner, because I'm using the 4 times objective, then this could also mean that this is actually magnified 4 times more than in real life, which it is not. Yeah, because there are 2 different types of magnification. One is called linear magnification, the one is just called magnification. Because actually this image, as you see it on the screen, how much it is magnified depends on the size of your monitor as well. Yeah, so this is again a different type of magnification. So this is kind of a little bit misleading, and this is why it's sometimes difficult. Yeah, I mean, if you want to, I can actually illustrate this to you. Just a second to kind of do it like this. Yeah, yeah. The actual image that my microscope camera delivers is this. This is basically the full image that the microscope camera delivers. Yeah, but maybe you are going to have a camera system which basically at the same objective magnification maybe only produces a smaller region from the center. Right, so you see, this is why the whole issue with magnification is a rather a difficult thing for comparison, right? So what I have done, let me just make this again. Yeah, return this back to the to the original one here. So essentially, what I've therefore done is I just show you always the stage, and now you kind of know it's 4 times. I know it's not always indicated 10 times. Yeah, but then again. Yeah, it's also not the same way that I see it through the microscope. Yeah? Because when I look through the microscope, I'm seeing again a different field of view. Yeah? So this is a little bit the problem that I'm kind of having, with communicating magnification, because whatever I say actually might not be, how do you say, meet the purpose. Yeah, so this is a little bit of an issue here. Yeah, a question, please. Give us the name of an affordable microscope with dark field. I suppose you mean DIC system and polarized colors, please. Affordable. As soon as you go to DIC, it's probably not affordable anymore. So I would say that many hobbyist microscopes that are out there with a condenser, you can easily convert to dark field. You can also easily convert it to polarized by adding to polarization filters. There are do-it-yourself adaptations that you can do. But DIC, which I suppose that you mean, which is the one that I have, is essentially you need to talk to high-end companies. Yeah, I know that the company Euromex has a microscope that basically has DIC. Olympus, of course, Nikon, Zeiss, they all have it, but that's getting expensive. Okay, so what I recommend generally for hobbyists, if you want to get similar images like in DIC, is to use so-called oblique illumination and color filters, and you'll be surprised of what you're able to get. Yeah, what's affordable? Again, microscope buying is a little bit, how do you say, the opinions go apart very much. I will just, if you just read the forums a little bit. I in the past have been recommending microscopes for hobbyists around, let's say, 300, 400 euros. Yeah, dollars, euros. There is no limit further up, and but generally, if you say, okay, get a microscope like this, then some people are going to say, well, actually, no, you shouldn't. You should get yourself a used microscope of a brand company. Then other people, again, have a different view. So there are so many different views on this. And I always say, if there are multiple conflicting views on a certain issue, probably then it becomes a question of taste, depending on what your expectations are. So I've made in my other YouTube channel, I've made some reviews. I've in the past recommended some models of Swift, the company Swift. Euromex, a European company from the Netherlands, has excellent microscopes as well. Full range, so you might also want to check out their website. But a lot is a question of personal preference as well. Okay. So when on your local time are you streaming? This local time is 21:30. So basically 9:30 in the evening, my local time. You don't have any scheduled program. Can you please change the time to 3 to 4 hours earlier, or Asians? Sorry, this is going to be super difficult for me. Okay, that is because during right now I've got holidays, but later on, if I do this a little bit earlier, it might actually conflict with my work. Yeah, because I sometimes return quite late from work. So I understand this problem. I do, however, hope that because the live streams are also recorded and basically in YouTube and can be watched pretty much any time in the future, I hope that yeah, you're not going to miss out on anything. But I know that this has been also an issue, but unfortunately, it's time-wise very difficult for me. Yeah? I wonder what would happen if I combined the zoom of a Nikon P1000 and the zoom of a microscope for a white blood cell. I suppose the Nikon P1000 is a camera. So basically, you know what? I'm going to just show you something. Okay? The whole issue about zooming. I don't know. You know what? I'm just going to use the. Who cares? just going to use the frog one again, just because it's easy to see the nucleus. And I just want to show you now. Scope view. It's a little too bright here. Is this the frog? Yes, it's the frog blood with the nuclei. Okay, so let's do the following. Let's go up here. Okay, I need to make it brighter. Here it is, right? So what I've been doing right now is I've been changing the magnification using the objectives. Okay, so a little bit of microscope question is why can you not make zoom objectives? No, not going to work. The distance is so small, and if you make now zoom objectives with a continuous zoom, you're really going to destroy the image quality. But you know what? People say, well, what's the big deal? Why can you not just add a zoom later on? Okay, just afterwards, like in the camera, for example, yeah. And you know you can, but you're not going to gain anything. I'm just going to show you now over here. If I just where is this? Here it is, right? What I could do, you know what? I just go up with the magnification. I'm just going to stretch it more and more and more. Yeah. Of course, it's going to be bigger, but you're not going to see more. And the reason why you're not going to be able to see more because we've already reached the limit of magnification of resolution, right? It's going to be bigger, but it's going to be more blurry. Right? And so that's why there is essentially, if you want to increase the magnification. There are many ways to do that. I mean, I can zoom in as a million times if I want to, but I'm not going to see more details. And that is essentially something that I want to inform you. If you want to get a good magnification, you have to basically do that over the objectives because the objectives are the limiting factor, right? Yeah, you can of course later on zoom in as much as you want, but yeah, you're not going to, just to illustrate this, you know what? Let's go down here to the 4 times, okay? Yeah, so this is the 4 times, remember, right? I mean, what I could do, let's go a little bit higher. Let's say the 10 times here. Okay. So what's the point of actually switching? What's the point of switching magnifications using the objective? If all I can do is just basically make it larger like this, right? Yeah, of course I can do that, but you're not going to see more, right? Yeah, so if you want to increase the magnification, you have to, you have to do this with the objective, yeah, because then you're actually also able to see more clarity. Yeah. So that's a little bit the thing here that I would like to emphasize is you have to kind of move away a little bit from magnifying over the camera and towards magnifying with the objectives. So let me, but I encourage you in any way to try out because some people have also said, contacted me and said, well, yeah, it's correct that you might not gain any more additional image detail when you zoom in, but still it might be more convenient to look at the object because it's simply bigger. So you just get it bigger and blurrier, but it's fine. Yeah, you know it's blurrier, but it's more convenient. Yeah, I get it. Yeah, the image in the thumbnail is troubling. Stacked cells, yeah, that's called rouleau formation. And I showed it to you as well. And that's a completely normal thing that happens when blood starts to dry out. And if you now do a little bit of research, you're going to see, wow, rouleau formation, the stacking of cells could be the sign of certain diseases. Yeah, but this happens in any case. Yeah, you can only do that, use this as a diagnostic indicator in the highly standardized conditions which we're not able to have. But sooner or later, pretty much all of the cells are going to start to stacking up, and as the blood starts to dry up and coagulate. So that is a completely normal process that happens. And sometimes, essentially, they don't stack up if I use a very little blood, because then they cannot stack up because they're compressed between the cover glass and the microscope slide. So, from Illy, do you think that the Swift 350 trinocular model is a good beginner's microscope? You see, again, I don't know. I think it's the one with the condenser. I think, why not? These are just standard microscopes, and my general suggestion is start with a low-cost microscope, experiment around with it. You're going to outgrow it maybe, right? Then you can always get yourself a better model. Yeah? Generally, what I recommend is not to invest too much money at the beginning, but this is, again, something that, like I said before, it's a question of taste. And some people are going to say, oh my gosh, why would you buy that? Buy yourself a used Zeiss standard microscope, a used one from eBay, and put it together yourself, because that's much better, right? Yeah, and I say, okay, do you really want to take the time to actually find a microscope like this and refurbish it and fix it and do all these things? It depends a little bit on how you define your hobby. Yeah, and what I try to always emphasize is it should not be so much the equipment. It should be more the observations that you're doing. Is this DIC microscopy? Yes and no. Many of the things that I have shown you today, especially during the stained ones, were actually in bright field. So this one here is in bright field, okay? And I'm just going to show you how this looks like in DIC. Let me turn up the light intensity a little bit more. Regular bright field, and that's now in DIC. Yeah, and you're going to see there is not so much huge difference in this case here. Yeah, so it depends really also on the specimen itself of whether DIC microscopy is useful or not. Yeah, but I can simply flip out the prism. Yeah, and then I'm in bright field. And today most of the things I've been doing in bright field because I did some staining. Did you add an anticoagulant to make the time-lapse video? No, I did not. How long was the original recording? Okay, the original recording was several minutes long. I just show it to you. You know what? This is actually the original recording. Okay, here, this is the original recording, not in time-lapse, and it says here 7 minutes, 48 seconds. Look, I can actually speed it up. So there's this little thing that I can drag, which you're not able to see, right? So this is, so it's 7 minutes. Yeah. I did not add an anticoagulant. Adding certain substances probably would or might probably impair the movement of the cells somehow. And the reason why it did not stack up or coagulate is actually quite easy, simple, because, where is the arrow right now? Here it is. Okay, is this because I used so little blood that the cells were not able to stack up or move around very much, because they're kind of really compressed between the cover glass and microscope slide. And this also means that the white blood cells here were only able to move in one plane and not also vertically. So they always stayed in focus. Because my first time lapses, I used a little too much blood. I was able to find some of those white blood cells as well, kind of hiding between the red blood cells, because there was so much, many red blood cells, and they also moved vertically, and then they were in and out of focus, and I could actually not see them moving quite well. So this one, no coagulant, just regular blood under the microscope slide with a lot of experimentation until I got the right amount of blood, and I looked at the right place on the slide, so that essentially the blood cells were scattered apart properly. Okay. What microscope am I using? I'm using an Olympus microscope. Yeah, it's a BX53 microscope. I'm from North Carolina and just started the 7th grade. Okay. Is there a Hay infusion for Paramecium, Aurelia, and Boseria? Okay, Hay infusions, big topic. Not so happy. Okay, So that is the thing that I would like to explain to you. What is a hay infusion and why am I not so happy with a hay infusion? And I'm going to give you a better tip. Okay. So generally, hay infusions have been used back in the day, 19th century maybe. Okay. What they've done is they've taken dry grass, added with water. Dry grass is hay. You add it with water and then you let it stand. And basically, what you then had, if you were lucky, were lots of, or sometimes, and then you add some pond water to inoculate the whole thing. And then what you then had is you had the growth of a whole range of different microorganisms in there. They called them infusoria, which is a collective term for vorticella and I don't know, all sorts of ciliates. That's why they called them infusoria. In German, Infusorien, because as a collective term for all of these microorganisms, because they made it from a hay infusion. However, and that's why I'm a little bit not so happy with hay infusions. The hay infusions basically have a lot of bacterial growth, especially if you take a large jar, okay, and you put hay and water in it. It really has a high concentration of bacterial growth, so it's possibly not healthy. It's kind of like you're decomposing this material. And second, because of the high concentration of bacteria that you have in there, they're using up all of the oxygen. And this basically means that Paramecia and other ciliates, which need a lot of oxygen, essentially cannot grow well. So if you make a hay infusion, you have to make sure that there's really good exposure to air, and so not too much. And I'm not happy about it because there are lots of bacteria in there growing as well, right? And what I would kind of suggest, or this has been done as well, you're still growing unknown bacteria, but what you can do is you take a pond water sample and you add a crushed grain of wheat, a wheat grain. Okay, you take 2 spoons, you crush a wheat grain, and you take this wheat grain and you drop it into the pond water sample. And then what you're going to have is you're going to have bacteria growing around the wheat grain, and then you've got also Paramecia and other ciliates feeding on those bacteria. So when you take some tweezers and carefully lift out the wheat grain after about 3, 4, 5 days, you might be quite lucky to find a lot of Paramecia in there. Okay, it's still bacteria that you might want to be careful. So but generally better than a hay infusion which contains even more bacteria, unknown bacteria. Yeah. Is not ethylene blue, methylene blue cancerous? It's not cancerous. It can so basically it can also be, or it has also been used for, I think, for disinfecting purposes even, but maybe it should, no stain is healthy, okay, because these are chemicals that react with your body and with the cells, so none of the stains is actually healthy. And methylene blue itself is has also been used and can also be available, for example, for treating fungal infections in aquarium fish. So what you do is you buy some of this methylene blue and you put it into your aquarium, staining the aquarium blue, and this actually attacks then some of the parasites and some of the fungi that might be growing on the fish. And then you have to exchange the water, of course, because you don't want to have blue aquarium water. So methylene blue has already also been used for medical treatment, at least in animals. So if it were really cancerous and really dangerous, then I would not be using it. Okay, but it does interact with DNA and does stain DNA, so you have to be careful, obviously, because anything that stains with DNA might be, also cancerous as well. But methylene blue, to my knowledge, is not. Otherwise, I'm not getting it on my fingers anyway. I've been working in the lab with ethidium bromide, and that stuff is really cancerous. Okay, carcinogenic. So you had to use double gloves and really you had to be extremely careful with that. Yeah. So what I'm going to do now is I'm going to quickly go through some possible remaining questions that there might be on here. Are there any hay infusion plants that I can use to get Paramecium plants? Well, hay infusion plants, yeah, it's just dry grass, but as I said, you got to be a little careful with this stuff, okay? Okay. Would you please say when and how long ago the first time you looked at your own blood with a microscope? Have you noticed a difference since then? No. I tell you, the last time I looked at the first time I looked at today was the last time. First time I think a couple of years ago when I started to make those microscopy videos. I see a difference all the time because the thing that causes the blood to appear most different is the amount of blood that they use, the thickness of the blood. The thickness of the blood layer beneath the cover glass, whether I look at the periphery of the cover glass or in the center, because on the sides it starts to dry out faster. So it always looks different, but it's not because of the blood so much. It's more about the preparation. Yeah. So that's simply the thing here. Okay. Yeah, exactly. The Giemsa stain is the common one that's normally used for blood. Yeah. The scale bar. Yes, that is also a thing that concerning the magnification again. And the reason why I'm not using this, especially for my videos, is because sometimes I make a video and then I zoom in more so that the cell or whatever I want to show kind of fills the screen or the video nicely. So I'm adding some additional zoom. And this would actually mean that I would have to actually calculate a new scale bar every time. Yeah, and yeah, so this is something I also didn't want to do, yeah, for time reasons. And then it basically tells you that this one is, I don't know, 2.87 micrometers. What does this tell you, you know? So for this reason, I said is yeah, simply to ignore those scale bars as well. Yeah. Why do veins tend to collapse but arteries not? It refers to that's actually an easy one to answer because the arteries have a much thicker wall. Okay, so veins are very flexible. You sometimes see them on the surface. You can easily compress them. Much thinner a wall. Why much thinner? Because they carry blood of a lower, significantly lower blood pressure, so they do not have to be so thick. Yeah. So hi, Microbehunter. I'm from the US and just started the 7th grade. Okay, sorry, that was the question that I just had. Okay. Okay, some. Yeah, my new hobby is buying old microscopes. Just like to have them. Yes. Yeah. Some people actually, yeah, especially for some old brand models, Zeiss, Olympus, Nikon, yeah, from the I don't know 1970s, 80s. Sometimes there is still quite a lot of second-hand equipment available. Apparently, the Zeiss standard model was manufactured in crazily amounts of very high numbers, and there seems to be lots of spare parts still available. And some folks actually like to assemble their own microscopes this way. Yeah. Do you know if caffeine has any effect on the white blood cells or the immune system? I know that caffeine has an effect on, apparently a mutagenic effect on bacteria, and definitely an effect on the nervous system. On the immune system, not that I know. Generally, many substances that you add in sufficiently high concentration will have some kind of an effect. Yeah. Oh yes, very cancerous ethidium bromide stain, yeah. What are the cells in the gut that do a similar job as the white blood cells in the blood system that are cleaning up? The cells in the gut. I don't know yet. It could be, it could be that some cells in the digestive system, I have to think now, in the villi, you've got the goblet cells that secrete mucus, you've got... Yeah, I can imagine maybe there are white blood cells also around there that are in the mucosa that do some cleaning up maybe. Sorry, for this one, unfortunately, I don't know the answer. Okay, so yeah, what I would like to say is the following. It's again 1.5 hours. I would like to simply call it a day again. Yeah, and I would like to wish you a nice week. I hope that today's video where we looked at blood, again, this is frog blood here, that was interesting. Yeah, and I think I'm going to be thinking of a new topic again for next week. I wish you all the best. Happy microbe hunting as always. And if you have some remaining questions that you were not able to post yet, please remember them. And so next week, if you type them in, then I'm going to try to answer them again. Okay, so but for today I will call it a day because it's quite late at where I am. It's 11 o'clock at night, and I'm quite sure that many of you also have, as already mentioned, quite inconvenient times, unfortunately. But I'm going to call it a day today. I wish you all the best. See you around next time. Happy microbe hunting, and bye-bye.